Thank you for the kind invitation. I'm really glad to be here, despite the fact I can't really, well, I see you now, so great. So what I will talk about is actually our work, what we are doing in Strasbourg. We are especially interested actually in studying mosquito and pathogen interactions, whether they are parasites or viruses. And what we try to understand is the molecular interactions between these vectors and their pathogens. And from this information, we try to design novel strategies that could be directed to limit disease transmission by these vectors. And today, what I will do is to actually talk about these two themes. So and this is all around one protein called saglin. So if I move to the next slide, well, I hope that can work. Do you see my...
Host? I don't know. Oh, it should be working. It should be working, actually.
It's just from my computer. I will try and stop sharing and start again. Actually, do you see my...
Yeah, we can see you can see your pointer, yeah, clearly.
Okay, great. So I will stop sharing and start again to see if it works better. I just can't change my slide.
Okay. Otherwise, you do a TED Talk without slides.
It would be a bit difficult, especially maybe on site would have been feasible, but from far away, that's a bit more of a challenge. Okay, let's try again. Share.
Let's see. Hopefully that works.
Let's do this. Yes, perfect.
Right.
So this is a very schematic view of what happens to a parasite when they enter a mosquito vector. So when a mosquito female will take a blood meal on an infected person, infected with malaria, she will take blood in her gut, and inside the gut there are gametocytes. These gametocytes will undergo actually fusion, where they form gametes, they fuse, form an ookinete that crosses the midgut epithelium. Then they round up, form a cyst that will develop over 7 to 10 days and will release lots of parasites in the body cavity. Some of them will make it to the salivary glands. And if this mosquito takes another blood meal, then it will transmit malaria parasites. Now, if you look at the journey of the parasites throughout this mosquito, it's actually far from a child play. And there are massive losses in parasite numbers that occur early on after infection. So this is shown here, where the parasite numbers is shown on the y-axis and the days after infection shown on the x-axis. So there's a sharp decrease in the number of parasites after infection, early on after infection. Then there's a multiplication of the parasites, large proliferation, and then again a decrease because not all the sporozoites that are released in the body cavity will make it to the salivary glands. And one of the genes we've been especially interested in is actually called saglin. And why? We've been interested because in quite a few years ago, actually, the laboratory of Marcelo Jacobs Lorena in the USA has published a nice paper suggesting that saglin was actually a receptor at the surface of the salivary glands, and this receptor was important for sporozoites, so the form that floats in the body cavity, to detect the salivary glands and therefore to infect the salivary gland. So, and in addition to that, they had knocked down the expression of saglin in the mosquito. So the mosquito had decreased saglin expression, and there was also a decreased number of sporozoites in the salivary glands. So we thought, well, this is interesting. Let's first of all make sure that we can reproduce this phenotype. And if this phenotype is correct, then we can maybe use this or exploit this as a way.
Wait, Stephaine, we have a problem of sound.
Here I describe the saglin locus in the genome of the mosquitoes. The saglin gene is localized in gray here. And what we've used is basically a CRISPR-Cas9 strategy to insert inside the Saglin locus another cassette that would break down the Saglin gene, and instead having these mosquitoes express actually GFP either under the 3p3 promoter, which is an artificial promoter expressing, driving the expression of the gene in the nervous system of the mosquito. This is just for us to know what mosquitoes are transgenic or not. And if you locks out this 3p3 promoter, then the GFP is expressed under the control of the Saglin promoter. And if we do that, what we observe is something that was known already, that Saglin is expressed only in the female mosquitoes, not in the male mosquitoes as shown here, and only in the salivary glands. And so you could say, so this is also shown here and dissected salivary glands. And you could say, okay, that's just GFP expression. Maybe you haven't detected it everywhere. So we did in parallel a Western blot, and that confirms we find Saglin only in the salivary glands. It's absent, obviously, in the Saglin knockout mosquitoes. You find it in the Saglin, in the wild-type mosquitoes, only in the salivary glands, and not in the midgut of the wild-type mosquitoes, especially neither in the carcass when there's no salivary glands, and neither in the hemolymph of the mosquitoes. The hemolymph is kind of the blood of mosquitoes. So clearly, Saglin is only entirely expressed in the salivary gland of mosquitoes. So we said, great. And then let's have a look at the phenotype. So if it's according to Marcelo Jacobs-Lorena's model, Saglin is expressed in the salivary glands, and it helps parasites to invade the salivary glands. So normally in the early days or early steps of infection, there's no effect. But this is actually something that was not true when we did the experiments with the Saglin knockout mosquitoes. So Saglin knockout is here, the control mosquito is here. And what you have here is a group of dots, and each of these dots actually is one mosquito. And what we count is the number of parasites that develop in the midgut of the mosquito. So what you can first see is that there's clearly fewer parasites developing in the Saglin knockout compared to the control mosquitoes. And in addition to that, there is a sharp decrease in the prevalence. The prevalence is the percentage of mosquitoes that are infected by parasites. So basically here, there are 65% of the mosquitoes, the wild-type mosquitoes, carry parasites in their gut, while there's only 13% of the Saglin deficient that can carry parasites. So this infection was done with Plasmodium falciparum. We did it also with Plasmodium berghei, which is a murine malaria parasite that we use as a model. And the reason is that we have plenty of different mutants for Plasmodium berghei, so it's very easy to work with. In this case, there's no decrease of prevalence because we reach very high number of parasites, and therefore probably we saturate the system. Still, we do observe a sharp decrease. It's 5 to 10 times less, fewer parasites are developing in the mosquitoes that are Saglin deficient compared to control mosquitoes. Now, obviously, we then looked also in the salivary glands, and in the salivary glands, we also see a decrease in the number of sporozoites that make it to the salivary glands. However, if we make a ratio between the number of parasites, the sporozoite in the salivary glands over the number of oocysts, actually, they are very similar in the control and in the Saglin deficient mosquitoes. So this means that if we have fewer sporozoites in the Saglin deficient mosquitoes, it's only due to the fact that they have fewer oocysts developing in the midgut. And it's not that these sporozoites that are released have more difficulty to penetrate into the salivary glands. So our data actually contradict to some extent the model that has been published by the Marcelo Jacobs-Lorena's lab. Still, this is very interesting, and that led us to ask an important question is about how a protein expressed in the salivary gland can be efficient to limit parasite infection in the midgut. And this came through a Western blot that was very simple. We looked at the presence of Saglin before and after a blood meal. And so when a mosquito is unfed, so it's a wild-type mosquito, obviously, unfed, there's no Saglin in its midgut, in the blood bolus that is inside the midgut. But if you dissect this same mosquito after a blood meal, then there is actually Saglin appearing in the blood-fed mosquitoes. So how does Saglin reaches the midgut? Very simple. Actually, when the mosquito bites, it will inject saliva and therefore also Saglin. And upon ingesting blood, it will actually re-ingest saglin at the same time. And then that's why saglin finds its way into the midgut of the mosquito. So the model currently is that saglin is not important for sporozoite entry into the salivary gland, but more for parasite development in the midgut. And this is typically what we work on to try and figure out how. Now, another question that was very important for us was to figure out if these saglin deficient mosquitoes that overall carry fewer parasites would be as good as the wild type to transmit malaria parasites, or preferentially not as good. And that's what is shown here. So if we make a parasite infection that is very high, then so in this case, mosquitoes that were wild type or saglin deficient were infected on a mouse. We let the parasite develop up to the stage where they are located in the salivary glands. And then we made these mosquitoes bite new naive mice. And then we measured, or we followed the parasitemia in these mice and figure out whether or not they would get infected. So if if the mosquitoes have been fed on a high burden of parasites, then all of them will transmit, whether or not they are saglin deficient. So that's shown here. However, if the mosquitoes have been fed on a mouse that with a low parasitemia, that's typically what we observe with falciparum. There's a decrease in the prevalence, and therefore most of them actually won't transmit, and most of the mice that were bitten stayed uninfected. There's just 1 out of 4 that got infected, the rest was not infected, in contrast to wild-type mosquitoes that all transmit malaria to mice. So typically, the absence of saglin can prevent transmission of the disease in low infection context, which is actually what happened for Plasmodium falciparum. And important other question is whether these saglin deficient mosquitoes were more or less fit compared to the wild-type ones, because if you kill a gene in a mosquito, obviously it can make it more susceptible, or more can have a fitness cost that would be less make its life less easy, basically, in the field. And what we've done here is to follow the percentage of mosquitoes that carried the saglin knockout allele when they were mixed with wild-type mosquitoes. So we crossed them and we followed them over 10 generations. And what you would expect, according to our cross, if it was regularly inherited or with no fitness cost for the saglin knockout, it's about 75% of those carrying the saglin knockout and 25% of wild-type mosquitoes. And that's typically what we observed, which means that basically there's no fitness cost of the saglin knockout, at least in lab conditions. Sorry. So basically, these 2 points are very important because, first of all, they show that basically there are 2 important points in terms of designing a strategy to limit parasite transmission, because they can indeed limit parasite transmission and they don't have any fitness cost. So that makes them, that makes the saglin gene a very interesting gene for transmission blocking strategies. Now, there are some remaining questions as well. Is how does saglin promote parasite development? That we still don't know, although we are working on. And also, if saglin is dispensable for mosquitoes, what is its role really compared to other genes that are produced in the saliva? Okay, so now I will move to actually how we can make use of this knowledge that we have acquired using gene drives. So I'm not sure every one of you is familiar with a gene drive, so I will explain you what it is. And our gene drives are actually based on CRISPR-Cas9 that I guess most of you know of. So a gene drive is actually an egoistic genetic element that will have a tendency to spread in the population at a much higher rate than a regular Mendelian inheritance. So how does it work? Imagine you have a gene drive cassette here that is shown in orange on a 1 chromosome, and this is the homologous chromosome. So upon meeting these 2 chromosomes during meiosis, when you can have actually pairing of the chromosomes, and if you express Cas9 and the guide RNA at that time, it will cleave. So the guide RNA is what targets the Cas9 nuclease to a specific site. So if the guide RNA is designed to cut the wild-type locus, the Cas9 will cut it and make a double-stranded break, and this has to be fixed. And if it's happening during meiosis, and especially during chromosome pairing, in this case, the way the cell will fix the problem is by actually copying the homologous chromosome into the chromosome that has been cut. And so you start with a heterozygous chromosome and you end up with a heterozygous cell and you end up with a homozygous cell. So if I think about in terms of inheritance, especially if this occurs in the germline, which is actually what happens, then without any gene drive, if I have a transgenic mosquito carrying heterozygous transgenic mosquitoes, carrying just one copy out of two different alleles, then I cross it with a wild-type mosquito, and only 50% of the progeny will be carrying this original transgenic cassette. Now, if instead I have a gene drive cassette, because my mosquito, my heterozygous mosquito, at least the germline cell of this mosquito become homozygously transformed, then it's actually 100% of the progeny that will carry the transgene, and therefore it gives to this gene drive cassette an enormous selective advantage, because instead of spreading at 50% as it is for a normal transgene, it will spread ideally at 100%, and therefore can very rapidly invade a target population. Okay, so that's the principle of gene drive. Now, how can we apply it to try and limit the transmission of diseases by vectors? So one first way is to try and limit the vector population. It has been very successful through insecticides and so on, that have been very successful at reducing disease transmission by mosquitoes. And so if there's no mosquito, obviously there's no transmission. And for this, you could, for instance, target a fertility gene in females. And if the females become sterile, then at some point the population will crash. Now, this is not what we would work on, because if the mosquito species is endemic, it has also its role in the ecological niche, and that might be actually perturbating and creating even worse situation. So what we work on in this specific case is actually a population modification. So in this case, what you would put in the gene drive cassette is something that would make the mosquito more resistant to malaria parasites and therefore less able to transmit the parasite. And the way the design I will mention in my following slides is actually what we designed, a dual gene drive, where there are two strategies that are coupled in just one gene drive. The first one is to disrupt a gene that is important for the parasite to develop, that's saglin we've been mentioning. And the other one is to express, it's coupled with a second strategy to express an exogenous antiparasitic factor. And you will see that actually these two strategies, or these two strategies target the parasite at different stages. One of them in the midgut stages and one of them in the sporozoite stages, so more the ones that invade the salivary glands. So I will explain to you what antiparasitic or exogenous antiparasitic factor we've been using, and then I come back to saglin afterwards. So we've used actually, we've been interested in an antibody. So mosquitoes do not produce antibodies to defend themselves against pathogens, but there is an antibody, a mouse monoclonal antibody that is known to bind the CSP, which is a circumsporozoite protein that is expressed at the surface of the sporozoite in very large quantities. And this antibody binds to it and has been shown to have two advantages. The first one, it would limit normally the salivary gland invasion, and second, which is more prominent, is actually prevents the sporozoite to move well in the skin and also to infect hepatocytes once it's transmitted to the next host. So this is the gene we wanted to make mosquitoes express. And the second one is, where do we make it express? And we decided to have it expressed together with a fat body protein called lipofurine. Why? Because first of all, it's an essential gene. Second of all, it's also highly expressed and it's secreted in the hemolymph. And I told you already that the sporozoites will bath into the hemolymph at some point in their life. This gene is actually, or this protein is actually important to transport lipids from the midgut to the tissues where it's needed in mosquitoes. And it's constitutively expressed. So the design is the following. This is the lipophorin gene here on a regular wild-type chromosome. And what Eric Marois in the lab has designed is this specific chromosome here, specific allele, where basically he recoded the signal peptide, inserted the antibody called 2A10. Okay, so I will call it 2A10 in the following part of my talk. 2A10 antibody. Then he used the big intron here to insert a marker so that our mosquitoes would express GFP and be easily noted. And then that's the rest of the protein. And because obviously we don't want to have the first part of the lipophorin gene to be bound to our antibody, we want them separate, then we introduced actually a cleavage, a proteolytic cleavage site that is the same as the one that is already present in the lipophorin gene. And this is just automatically or naturally cleaved in the Golgi. Okay, so what happens in a wild-type mosquito? There's two forms of these two cleaved form of lipophorin that are produced, so lipophorin 1 and lipophorin 2, and P. falciparum floats in the hemolymph together with the lipophorin. Now, in a transgenic mosquito, in this case, there's obviously the two lipophorin, but in addition, this antibody that can bind to Plasmodium falciparum, okay? And in addition to that, as I told you, when my mosquitoes carry this specific 2A10, lipophorin 2A10 allele, they are green, and therefore I can sort my mosquitoes according to their fluorescence. And I know if they have one or two copies of the transgene. Okay, so that's the way we have designed the thing. The next part was to figure out again if this transgene was let's say, had any fitness cost on the mosquitoes. And see, this is again followed over several generations. We follow the percentage of mosquitoes carrying that are either homozygous for the lipophorin 2A10 allele, heterozygous or wild type. And you see that with time, there is a decrease in the percentage of mosquitoes carrying this allele, which means that there's some fitness cost, but it's not tremendous. So the next step was to see whether these mosquitoes, so homozygous mosquitoes, having this expressing this antibody would be less good at transmitting malaria. This is shown here. So again, the same strategy as what I've shown you for Saglin before. We infect wild type and lipophorin 2A10 mosquitoes with parasites. We let them arrive to the salivary glands, and then we infect mice on these mosquitoes. And for each of these mice has been bitten by 10 of each of these mosquitoes. So some of them have been bitten by 10 wild type and some others by 10 GFP plus mosquitoes expressing the 2A10 antibody. And what I show you here is that if the mice, were bitten by wild type mosquitoes, most of them, so almost 100% of the mice get infected. However, if they are bitten by homozygous mosquitoes expressing the 2A10 antibody, in this case, there's a decrease by 70%. And this is more or less the same when they are bitten by heterozygous mosquitoes, meaning that, just a little bit of the antibody is actually enough, or even if the quantity is a bit lower, it's still very efficient at decreasing transmission. And I just show you a control here, which is here, it's actually Plasmodium berghei that we work with, but we've inserted in its genome the P. falciparum CSP so that we can infect mice. Otherwise, that's very difficult to make this experiment. And if the Plasmodium berghei CSP is not replaced, in this case, the mice that are bitten by wild-type mosquitoes or by the homozygously expressing 2A10 antibody, then they are just transmitting the same way. So it's very specific to the P. falciparum CSP. Okay, so what you have to remember from here is that when mosquitoes express the 2A10 antibody, then they are less prone to transmit parasites to the next host. And this is despite the fact we selected actually mosquitoes that had very large amount of parasites. Now, how to drive the 2A10 spread in the population? Because right now, there's no gene drive involved in this allele, the 2A10 allele I've presented you. And we've tried several strategies, but I will tell you the one that worked. So actually, that's where Saglin comes back. So what we designed is a gene drive in the Saglin gene. So the gene drive cassette, you remember, it's a cassette that has that will cut the wild type locus and insert itself by homology directed repair into the locus that has been cut. So in this case, this is the gene drive cassette with Cas9 and 3 guide RNAs that target the Saglin locus. And so when it's cleaved, then this gene drive cassette can be copied inside the Saglin gene. But in addition to that, we also inserted a red marker to follow our transgenic mosquitoes. So this one are red in contrast to the other ones I told you, which are green. And we also inserted 3 guide RNAs that target the lipofurine allele. So if in my mosquito, I not only have the gene drive, but I also have the LP2A10 allele, in this case, the wild-type lipofurine locus will be cleaved by the Cas9 and the guides that target this locus, and the lipofurine 2A10 allele can be copied inside. So my gene drive here with a single Cas9, I drive, first of all, the insertion of the gene drive cassette into the Saglin, but also the spread of the LP2A10 allele in the population. And now the question is how this combination of both Saglin knockout and LP2A10 expression will work on transmission. Same experiment as before with fewer mice, so it's a kind of a preliminary experiment, but still we see a decrease that is sharper than with the previous one. So from 70, we go down to 90% decrease. And again, here we did not, we were pretty harsh because we selected mosquitoes that were heavily infected, and therefore the Saglin knockout effect has been, probably sort of decreased in the selection we've made here to do this experiment. So probably the real decrease would be much higher than 90%. Okay, so with this, we then, the next question, if you make a gene drive, is to try and spread this a specific modification that make mosquitoes more resistant, is to spread them into the population of mosquitoes. So we did cage experiments where we crossed this mosquito carrying the gene drive, the Saglin gene drive and the LP2A10 allele. We crossed them with wild type and we followed their spread in the population over time. So here you have 8 populations where this has been created. Actually, there are 2 populations that have been done right from the start, and the population 3 to population 8 are mosquitoes that have been collected at some point during this process to start new populations in different strategies, but I don't have time to enter into the details. So remember, maybe here, just look at the allele frequencies. So the continuous lines, green is for the LP2A10, and red is for the Saglin gene drive, sorry, there. So you follow them and you see that in most populations, actually the green allele is fixed very rapidly. So it takes about 10 generations. So here it's the percentage, the allele frequency over the generations. You see about 10 generations, the green allele is fixed in the population. So it goes very fast. The lipofurine allele is fixed. However, for Saglin, it's a bit more, flexible to some extent. And sometimes you go down to 50% to go back up again afterwards. But it's basically more difficult for Saglin to be fixed in the populations. So how can we explain this? And this is through the generation of resistance, so resistance allele. By that, I mean gene drive or resistance allele to gene drives. So I told you about how a gene drive works. It's because there is a homology-directed repair of a double-stranded break that is created by Cas9. Okay, so double-stranded break, you have the donor DNA, which is usually the chromosome carrying, for instance, the gene drive that will copy inside, and then you have a gene drive. Now, there's another way to fix a double-stranded break. It's called non-homologous unjoining, and in this case, basically the cell will try to stick together the 2 bits of DNA that have been cut by gluing them, right? And the fact that it glues them means that it will add some nucleotides or remove some nucleotides. So at the end, you have a position that is modified. So the guide RNA cannot recognize it any longer, but it's not repaired by the homologous piece of DNA, right? So at the end, this is what we call resistance allele because they can't be targeted again by the guide and the Cas9. So that's typically what we wanted to measure. So we went back to these populations, and for Saglin, we collected the mosquitoes very early on, so in the first population, so in the population 1 and 2 at the early generation. And for lipofaurin, we sequenced the lipofaurin allele from these mosquitoes here rather later during the generation time. And so for Saglin, what we observe, so everything that is green here on this slide is modifications, okay? And what you can see is that there are sometimes we have when two guide RNAs have cut together, there might be a big deletion, but most of the time it seems that guide RNAs cut just once because they are like there are indels into these locations. And sometimes you can see also that indels, so you have one position here that is modified, one of them that's with a modification at the locus corresponding to guide 1, and for the other one there's no modification at all in the rest of the sequence. So it means that it's also a cumulative iteration of modifications over generations that accumulate over time. And these resistance allele basically become more and more resistant because at first they may be resistant to one guide cut, but now with time they may be resistant to actually the three cuts, three guides in total. And why is it accumulating in Saglin especially? There are several reasons, but one of them is the fact that Saglin is not an essential gene. So if you make an indel and Saglin is not expressed, it won't have a fitness cost on the mosquito and it won't be transmitted to the next generation. Now for lipofaurin, it was a bit more surprising to find a resistant allele, but we did find one. And for this one, there was no modification on guide 1, but there was one modification on guide 2 and one modification on guide 3. So for guide 3, the modification, was in the intron. So obviously the fitness cost is usually limited if it happens in an intron, except if it targets a specific position. But for guide 2, it was actually in the coding sequence, but in the signal peptide, and it's just 3 amino acids that were deleted. And so just one amino acid got eliminated, three nucleotides that were eliminated, so one amino acid, and at the end, it did not impair the function of lipofaurin. So we had one lipofaurin allele that was also created. So one of the problems is that if you have the resistance, the gene drive resistance allele accumulating, then after your gene drive doesn't work any longer. And so you would probably see actually a decrease in the percentage of this gene drive, these red and green markers in the population. The other thing that we looked at is whether the guide array was actually conserved. And what we've shown is that actually there were recombination because the guide RNA arrays are very repetitive. So there were actually quite a few recombinations that occurred. So altogether, and I see you, Cyril, so I will finish up there. So the take-home messages from my talk are the following. So first of all, we better characterized a gene called Saglin. And we've shown that Saglin, although expressed by the salivary gland, it promotes parasite development in the midgut, meaning that parasites are able to use, mosquito proteins to promote their development. We've shown that Saglin knockout can reduce transmission, and that it's also true when the 2A10 antibody is produced in the mosquitoes. So these two modifications can make mosquitoes less able to transmit diseases, and we coupled them into our dual gene drive strategy that targets, therefore, parasite at 2 different stages. The spread of the gene drive was actually very good for the 2A10 allele because it was about 100% of the population that was modified, a bit more difficult for Saglin, also because it's on the X chromosome, so only during females can it work. And also we found the appearance of refractory alleles for the gene drive and also recombination in guide RNA arrays. And this is typically things that we work on to improve the design of the gene drive to make them more efficient and less able to make refractory alleles. So yes, as I said, gene drive optimization, what are the perspectives? It's also to figure out how Saglin works. And also whether if we imply such a selection on the parasite, whether we would develop actually some resistant parasite to both the 2A10 antibody and the Saglin lack in the mosquito. And with this, I will stop here. This is the building where I work in Strasbourg, and people who've contributed to the work are in bold here, and we've have also some collaborators, including Eric Calvo, who provided us some nice reagents for the saline story. Thank you very much.
Thank you so much, Stephaine. It's actually very refreshing to be on the other side of the life cycle, not looking at the parasite only. Bravo for the tour de force in being able to do such modification in the vector and such complex analysis. I'll ask the audience whether there are questions. I do have a couple. Yes, please.
So nice talk. My question is, when you select the genes for gene drive editing, what do you know about that locus from the mosquito population? Is it very homogeneous, or is it a heterogeneous? Because you nicely showed in the end some of the mechanisms how resistance can come to evolve.
Yes.
But if there is a lot of heterogeneity in the population, then you would expect. So can you estimate the level to which resistance can occur simply by sequencing individuals from the population?
Yeah, so you're very right. Actually, the polymorphism in mosquito population is very high. And so what could happen is that we don't even need to generate resistant alleles to our strategies, because they already exist in the population and they would therefore be promoted, or their spread would be promoted. So one of the reasons is that that's one of the reasons why we selected 3 guide RNAs for each of the genes, because with 3 guide RNAs, you're less likely to miss, one site. Even if there's one polymorphism in one guide RNA, there are still 2 that would function. And second of all, there are good studies that have been led, not by us, but by Dominique Kwiatkowski, for instance, where they sequenced lots of mosquitoes, and then you can have access to this polymorphism in field mosquito populations. And so with this in mind, you can always try and design your guide RNAs in places where they are highly conserved. So, but you're right, this is one of the limitations for the spread of gene drives in mosquito populations. And what we wanted to do here was really a proof of principle of the strategy, and not necessarily, I mean, this is clearly not something that will be released in a field. That's clearly not. So
I was about to ask.
Yeah, and figure out what are the things that still need to be improved to some extent. Yeah.
So I'll carry on with this. So this is, you don't want to do a spread in the field. You don't want to release such mosquito in the field that's not in the pipeline.
Well, so not for the designs I've shown you. So this one is not good enough to not meeting the request or the requirements for such a thing. In addition to that, there would be, there are some legal, ethical.
Some others have done it already, huh?
Yeah, but there are a lot of things that have to be discussed in front. Our goal for us is to provide as much information as possible to the policymakers, and also the good part and the bad part about it, the limitations, the advantages and disadvantages of the strategy. And again, this may contribute to, if we are to eradicate malaria one day, this may contribute to it, but this is not going to be a magic bullet for sure. And so it's just one of the strategies that could be employed. So yeah, I would stay very humble on that side.
Do you think it's druggable? Not knocking it out, but maybe using a drug strategy against protein?
You mean for?
Using an insecticide or something that contains a molecule that will be targeted against agin.
I'm not completely sure, but maybe it would be possible. I don't know. I mean, right now it would be difficult to think about it, because I simplified a bit, but the promoter sequence of saline is actually complicated. It's not directly in front of the gene. It's actually upstream of a set of many genes. So actually many genes, 5 genes, I think, or 6 genes in total, which are all expressed in the saliva and are probably sort of redundant to some extent. So I I'm not completely sure it would be easy, something very easy to do, but why not? It doesn't seem to be essential. And now the question is, how would you make so would you
Or
spread
something
it?
blocking it, actually. I was thinking, something blocking the interaction between the parasite and that protein, actually, because it's
sure.
not essential, obviously. Yeah.
You could vaccinate people against Saglin, and then that could be maybe one strategy, and parasite would not be able to use it in the gut.
I have a question in the audience.
Nice talk. So how did you do the feeding assay? How you feed those mosquito? Like I think you say gibbericide. You are feeding gibbericide.
So for falciparum, we culture parasites in cell culture, like regular cell culture with red blood cells from humans. For P. burgei, we use actually mice. So,
Which strain of falciparum?
So we use a clone called A11 that has been produced by Archer's lab, but it's from NF54. And it's a high gametocyte producer compared to other, Plasmodium falciparum lines.
Long live India and France friendship.